All right, so let's wrap up this process of mitosis. Now remember, the whole cell cycle is interphase and mitosis combined. And interphase, we divide it out into G1, S, and G2. Mitosis then gets divided out into prophase, prometaphase, metaphase, anaphase, and then lastly, telophase. So an easy thing to do when we're looking at mitosis is to follow the DNA. What's happening to the DNA? We start with, let me get back, there, where you at? We start with chromatin in prophase. It turns into chromosomes as it goes into excuse me, prometaphase. It's still chromosomes in metaphase. As it goes into anaphase, it's going to become chromatids because it's ripped in half. And again, there's, depending on the book and the resource, you'll see daughter chromosomes and stuff. I'm going to keep it this simple. Chromatin to chromosomes to chromatids. And then as we come back, or not come back, as we move into telophase, the chromatids turn back to chromatin. And I'll get to that in just a second here. So let me finish. Let's get telophase. Okay, so telophase, remember, we've pulled apart the chromosomes into chromatids. Telophase, the chromatids unravel, fancy word, is decondense back into chromatin. The cell membrane finishes pinching in half. So those cells that they're pinched in half, they're half the size of the original. So let's go back to the drawing for a moment. Okay, so you started to squeeze during anaphase. The squeezing continues. There. Same thing on this side. Keep squeezing. Get rid of all this. Chromatids will not look like little V's. Instead, they go back to, here's our little spaghetti noodle. Pile there. Pile here. Those little things we called centrioles, those dissolve. We don't need them anymore. And a new nuclear envelope forms around the chromatin. Okay. So new nuclear envelopes form around chromatin, and cytokinesis finishes. Done. We have two cells, half the size of the original, side by side. So again, remember, chromatids turn back into, where to put it? Back into chromatin. Both cells sitting side by side, and then they would say, okay, we're right here. There's one, there's two. Let's both go into interphase and start G1, S, G2, and we'll do it again. 24 hours later, there will be four of us. This is the normal cell cycle, what all of our cells should be doing if everything is going correctly. But there's always the but things don't always work right. So here's where we have problems. Genes. Genes aren't the right code. Genes are inaccurate. We don't have the right instruction book. So here's an example. Normal cell goes through the cell cycle normally. There's a gene called the proto-oncogene that is supposed to control the cell cycle. Let me move this over. I'm going to squeeze this a little bit. Oh, I don't want to get too bad. Sorry. Okay. So the proto-oncogene, it's kind of a funny word to be using, but molecular folks came up with that. So the proto-oncogene is literally, let's think about it as the gas pedal for the cell cycle. So there's a growth factor that attaches to a receptor protein on your cell. So remember that breast cell that we looked at? That receptor attaches. So it attaches, stretch this, there's a little more room. All right. So it attaches to the protein on the cell membrane, throws a signal into the cell and says, grow, divide, make a copy. Yay, happy, healthy, normal cell. That's this path here. Ooh, oh, that wasn't supposed to happen. The path right here. Now you have a normal cell. Sometimes proto-oncogenes are mutated. There's a problem with them. They're overly active. So a region of your DNA is not the right information. And that gene, think about a gas pedal. It doesn't shut off, or a gas pedal that doesn't shut off. That oncogene is not taking its foot off the gas pedal, and that means the cell will copy. Get the idea? Keep copying, and there you now have a tumor forming because you have cells that are not regulated. They're not told to stop after they reach a certain number of divisions. They're not told to pause or anything like that. So literally the gas pedal signal stays on, and now their cells grow uncontrollably. So there's one problem, possible problem there. The other side of this little diagram shows us what we call a tumor suppressor gene. So it's a different gene. So if we want to simplify it, we could say, here's gene 1, here's gene 2. So your tumor suppressor gene is supposed to block cancer. Control it. Say, oh, hey, don't grow, stop. You're not doing it right. The tumor suppressor should block cancer development and literally tell the cell to self-destruct if that gene is on. One of the more notable ones is one called the P52 gene. That's the one that should be telling that cell, if you're developing incorrectly, stop it. We're going to self-destruct you, and you don't continue to develop. But if that tumor suppressor gene is not working, it's faulty. It's not accurate. It's like having a security guard who's supposed to check things if your security guard's asleep. stuff goes through that shouldn't go through. So your tumor suppressor gene, if it's turned off, it can potentially lead to a tumor forming because there's nothing stopping it, controlling the development of the tumor, regulating the cell growth, saying something's wrong here and we don't want this to continue. So these are issues where your genetics play a huge role in your potential for developing cancer. We can't control that. We do not have the ability to control these genes at this point. The first thing is identifying, do you have a risk of cancer because of your genetics? Then we now start taking steps. That's looking at your family tree, understanding your genetics. So, all right. So here's the P53 gene. I'm sorry, that should have been P53. I don't know why I was, let's see, erase P52, P53. That's my apologies. I got my number off. P53 gene. So there's where P53 comes into the picture. Cell cycle makes mistakes, there's damage. P53 can say, self-destruct. Get rid of it, it's not good. Or it can pause and repair. Let's fix. P53, think about that gene kind of like spell checker. If you guys are writing a paper for a class or writing a big essay or writing a big long email and spell checker goes in and says mistake, 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 you then decide, do you just scrap it and start over or do you fix it? But imagine if spell checker's not on and you submit that email or that paper with a bunch of errors in it. That's not what we want to do when we're making copies of ourselves. So hopefully all of our P53 genes are on, they're active, they're functional, they're working, and they can block mistakes and prevent these things from happening. But we don't know that until we start looking at our family tree. So, all right, so kind of the overview when we look at genes that stimulate and inhibit the cell cycle. So there's different genes involved here I want you guys to be fairly aware of. We got our tumor suppressor gene, we have our proto-oncogenes. So when that growth factor hits the cell membrane, it triggers events inside the cell. What is the cell going to do? Is the gene telling the cell, do this, make this, go down this path? Or is it pulling back saying, fix it, self-destruct? What's the job of it? What is the job? And then is it functional? Is it working? So that's a huge, get that out of there. That is a huge thing to be aware of, is our genes. So I want you guys to start thinking about your families, your family tree, and if our genes are functional. So that's why it's important to have conversations or to try to learn as much about your family health history, parents, grandparents, genetic connections, what's going on there. It can make a big difference, huge difference in overall awareness of this issue. So, all right, so what we're going to start looking at next is, how does this stuff spread? How does it move around the body? If you have a melanoma, is it a big deal? Do you just ignore it? If we do ignore it, and I don't care, what could potentially happen? What is the big long-term or potential consequence of just ignoring something like this? So we'll take a look at that in the next part.